All right, so let me start now with yet another lecture on systems genetic approaches. And there have been several lectures that described very sophisticated tools. And I will describe yet another approach based on structural equation models. And so the approach I describe is published in several forms. So in one article, Jason Ayton, who was a former doctoral student, developed the so-called network edge orienting software, which is based on structural equation models. Also, some of this material can be found in Chapter 11 of the Weighted Network Analysis book. And finally, I will describe an application by Chris Plaisir and Päivi Pajukanta to study familial combined hyperlipidemia. So what about this network edge orienting software? Again, it uses this basic idea that SNPs can be used for learning directed networks. And there has been a vast literature on that topic. So within the realm of genetic epidemiology, people have used that insight in an argument called Mendelian randomization for avoiding confounding issues. And but lately, of course, this has been rediscovered and extended by other authors. For example, Eric Schad published an article that has been cited several times. And just to remind you, there's this fundamental paradigm of biology that says sequence variation precedes changes in gene expression, which then may affect protein changes and may ultimately give rise to clinical traits and diseases. And often, of course, we observe correlations between these various variables, but the one thing we are very certain of is that SNPs are causal anchors in the sense that when you have a correlation between a SNP and a gene expression profile, then in most cases it means the SNP causally affected the gene unless there are design issues in your data or terrible things went wrong, you know. Now, just to give you an intuitive reminder of what an edge orienting problem. So imagine you have these chromosomal markers, SNP markers. You have various gene expression levels, various genes. You have some clinical traits or physiologic traits such as insulin levels or HDL levels. And often correlations tell you, well, there's a strong correlation, a strong relationship between various edges. And when you just have, when you only have a co-expression network, we really cannot dissect causal relationships. But what we hope to get as part of the solution of the edge orienting problem is edges that show directions. And in our application, we produce so-called LEO scores, which stands for local edge orienting scores. And a high LEO score is meant to suggest that we have strong evidence for this causal direction. For example, here the LEO score is 3.5, which would suggest there's strong evidence that this expression level is causally affecting HDL levels. That's what we want to accomplish. So the NEO software and also the R software that I will describe shortly takes as input a set of quantitative variables. So these could be physiological traits or clinical traits, but also, of course, gene expression levels. And another critical input is to our SNP marker data or genotype data coded in a numeric fashion. So when you, for example, 0, 1, 2, counting the number of the minor allele, but you could also have a recessive coding or dominant coding, but it will be important to have a numeric coding of these markers. Now, here I of course emphasize the use of SNP markers, but if you have other causal anchors, you could use them as well. And then the software and the R functions output these scores, the Leo scores, for assessing causal relationships between these correlated quantitative variables. And Jason Ayton has produced the NEO software that gives you this type of an output, where for each edge, for example, from trait A to trait B, the software calculates this Leo score, say 1.5, which then suggests that there could be a causal relationship where A causally affects B. So that's the hope. And as I mentioned, this NEO software and our approach is based on the evaluation of local structural equation models. And just to remind you, structural equation models have long been used in statistics. They were first described in the 1970s. It's a well-established tool for dissecting and evaluating causal models. Structural equation models are really a generalization of multivariable regression models. All of us know regression models, but of course people haven't stayed at that level. They've expanded it to these structural equation models. Oops. Now, in our situation, we restrict our attention to very simple causal models. So here I describe 5 different causal scenarios involving 2 quantitative traits, A and B, and 1 SNP marker. M stands for marker. And so we evaluate these 5 different causal scenarios. For example, this one where the SNP marker causally affects trait A, and then trait A causally affects trait B. That could be of great interest. But there are, of course, alternative causal scenarios possible. For example, the marker could actually affect trait B, which then affects trait A. Or the marker affects A and B simultaneously, and there is no causal relationships between A and B. And when you think about it, you could probably think of other causal scenarios, but our software can only distinguish these 5 types. And why? Because we found other causal scenarios are indistinguishable from one of these. Now this happens when you have a single SNP marker, for example. But also sometimes you have multiple SNP markers available, and then you can come up with other causal situations. But I don't talk about those today. The one thing to highlight is that my focus is on a single edge, meaning 2 quantitative traits. And earlier today you heard, of course, a talk by Bin Zhang, and you will hear other talks by Jun Su. And also yesterday, Brian Yandel and Elias Chaibunitu described causal modeling approaches that involved multiple edges, really networks, causal networks comprised of multiple nodes. And I have a much simpler scope and much more restricted scope here, In any event, when you have a causal scenario, then you can evaluate how well your data fit this causal scenario by using this structural equation model framework. And just to give you a glimpse of what is being done, it is that there is again a objective function that is being minimized. And so when you have a causal model, it gives rise to a certain parametrization of the covariance matrix, right? What do I mean by that? So we have a SNP marker, we have two quantitative traits, A and B, and we can form a three-dimensional variance-covariance matrix among those three variables. And so we observe this variance-covariance matrix. I mean, this is just literally output of standard the cov function in R. Now, what does the causal model do for us? The causal model suggests that this observed variance-covariance matrix has a certain form. And this form is parameterized. There are certain parameters. And so when we evaluate the causal model, we actually fit the parameters by minimizing this function. And just to be clear, these straight line indicate the determinant. So we take the logarithm of the determinant minus the logarithm of the determinant of the observed variance component matrix and plus trace and so on. And M is the number of variables. So the SEMR package then finds the optimizing solution. It fits the parameters. And then after plugging in these minimizing values for the parameters and multiplying it times the sample size, we arrive at a chi-square statistic. And this chi-square statistic evaluates the model fit. So if the once you have a chi-square statistic, and here you have the corresponding degrees of freedom, you can calculate a p-value. And this p-value tests the hypothesis that about model fit, and in our situation, we want to obtain a high p-value because it suggests that the causal model fits the data, right? A low p-value means that the causal model that you study doesn't fit the data. But in practice, of course, you have a causal situation in mind. You want that the marker affects trait A and A causes B. You have a causal hypothesis. And so for you, a high p-value is good news because it says the data that you measured really fit this causal scenario that you outlined. So I make this point because throughout biomedical research, we are always opening the champagne bottles when we have a small p-value. But, but not here. In this structural equation model setting, we want a high p-value. So to give you a few more details, remember I had these 5 causal scenarios involving one single marker. And what I omitted earlier is there are certain parameter values. For example, when you have a causal edge from M to A, there's a coefficient, a path coefficient, alpha. And also, there are certain variables that model noise. So there's epsilon is the noise variable, and so on. And also, there's a parameter gamma going from A to B. And the epsilon is a noise variance, so there's a parameter called sigma square measuring the variance. So although these models are very simple, they do involve certain parameters, alpha, gamma, sigma square. And as I mentioned, these parameters are being fit by minimizing this objective function. All right, and then for each model, we then calculate a p-value based on this chi-square statistic that I mentioned. And so we end up with 5 different p-values. And so in principle, selecting a causal model is rather straightforward because you could say, well, I choose the causal model with the highest p-value as the one that fits my data. It's as simple as that. However, looking at all 5 p-values can be a tedious exercise, you know. And so, therefore, we define a statistic which is a relative model fitting index as follows. So remember, we are often interested in one causal scenario. The SNP marker causally affects a gene A, and that gene A causally affects, for example, HDL levels. That's our causal situation that we're interested in. And we get a p-value for it. But we divide this p-value by the best fitting alternative, by the p-value from the best fitting alternative model. Remember, there were 4 alternative causal situations, right? And so for each of these 4 causal scenarios, you also get a p-value. And so then the p-value of the next best model can be obtained by forming the maximum p-value across these 4 alternatives, right? And so therefore this ratio of the p-value from your causal model of interest divided by the maximum over the 4 alternatives gives you a relative fitting index for evaluating the fit of your model. And then after you obtain this ratio, we transform it using the logarithm with base 10. So what does that mean? So let's say this local edge orienting score, this Leo score, takes the value of 1, then it tells me the following. It tells me that my causal model of interest fits the data 10 times better than any of the alternative models, you know. Now, intuitively, this is a good evidence that it's 10 times better than anything else, and therefore it may be worth exploring, you know. So to remind you how we go through a typical analysis. So yes.
Could you, in practice, get a result here where actually originally your null hypothesis was rejected for all of the models, but this fit makes it look like one is quite a bit better than the other 4, but really you've rejected the null, so do you have support for that model or not?
Yeah, let me repeat the question for the camera. So the question was, could it be possible that actually all 5 p-values are significant, meaning none of the 5 models fit the data? And then, of course, when you form ratios, it could still be that one model looks much better than the others, but actually none of them fit. And of course, that could happen, you know. So everything that can go wrong will go wrong, you know. That's Murphy's law. So yes, that can happen, and sometimes it does. And therefore, this really highlights that this relative fitting index is actually just the first step. So it allows you to quickly look at certain traits A that are semi-interesting. But then, after you find an interesting gene, for example, you then really want to go back to the individual p-values, look at them. But even that is not enough, because structural equation models really allow you to output multiple fitting indices. There are very famous fitting indices such as RMSEA, CFI. There's a vast literature on fitting indices, wall statistics, and so on. And so in our software and also the SEM's R package, which was developed by John Fox and his colleagues, they really output all of these fitting statistics. And no doubt you want to look at all of them before you publish your gene in a paper. It's really a fruitful exercise. Having said this, we have several applications where the Leo score produced rankings that were very meaningful, you know. And remember, every speaker in this room emphasized that these causal analyses based on stationary data make a lot of assumptions, you know. And so I think all of us are in agreement that whatever you come up with, always use biologic validation experiments. These are always just exercises in generating hypotheses. That's all it is, you know. Yes?
Yeah, just a small question is that
Yeah.
the LU index, what you are saying
Yeah.
here, test these different models.
Yes.
Similarly, we heard yesterday also about the causal model selection test, CMST, for testing those
Yes,
models.
yes, yes.
What is the difference between these two model testing methods?
Yes, so the question is, we heard about several statistics for comparing these different causal models. For example, the CMST statistic from Elias Shaibub-Nito and Brian Yandel, and also the AIC measure that Eric Schad used in his paper, and various other measures. And so I realize how your head is spinning being exposed to all of them, you know. But here's the good news. When you have this very simple situation of 2 quantitative traits and 1 genetic marker, I claim that we would hopefully all agree on which model fits best, certainly if there's a strong evidence for one causal scenario. And we have done some comparisons, and the results were very reassuring. So that's the good news for the simple models. When you come to more complicated models, the minute you have 3 quantitative traits, 4, suddenly things start to diverge. And so I don't want to comment on it, but probably the other speakers have the strong opinions on which method works better. Yeah. Yeah.
Especially in the situation, for example, like M to A to B.
Yes.
For that A is also dependent on some other variable like age and gender and
yes, yes.
etc.
Yeah.
Is there any way to accommodate that in this model?
Yeah.
To make it simplified and still use it?
Yes. So the question is, what if you have yet other confounding variables, for example, age and gender and so on? So there's several ways to include these other variables. One is to literally include additional causal edges. If you feel that age affects gene expression levels, you could include it. And then you could fit a more complicated structural equation models. Just to be clear, structural equation models can deal with very complicated causal graphs. And there's a vast literature on that, you know, so you could include it at that level. But also, some of us use tricks where we first form residuals in some shape or form, you know. Or you stratify the analysis. You fit a causal model only on young subjects, you know. There are all sorts of tricks one can use, you know. But let me proceed with the lecture. And so this gives you a sense of how we calculate these Leo scores. So now, among all the criticisms that you can raise about these causal inference models, the one thing you probably may not be aware of, that the Achilles heel is the genetic marker, okay? That's my number one criticism. As you know from human GWAS studies, you sometimes need tens of thousands of human subjects to find anything statistically significant. What does that mean? Often the SNP has a very small effect on the clinical trait and also on the gene expression levels. And I don't need to tell you, if you put garbage in, you get garbage out. So if you use causal anchors that you are really not sure of or who have a negligible effect, you can still calculate your causal scores, but things can go wrong. And so there's no, it's not a coincidence that most of these models have been applied to model organisms, for example, mice. Why? Because there we get hugely significant QTLs, you know, where we have more confidence in the causal anchors. But in human subjects, be aware of that limitation. So, for me, the critical step is to specify these genetic markers. And in our software, we have certain ways of carrying out SNP marker selection, but the truth, for example, based on a forward stepwise SNP selection approach. But the truth is, I'm most comfortable if you start with a SNP that was published in 3 different articles using multiple linkage analysis, GWAS studies, a SNP you really trust. start with, and therefore my emphasis today is on a method that uses a single SNP, because I'm pessimistic that you have multiple credible SNPs. So in any event, once you have this SNP, then the software allows you to look at thousands of pairings of genes and traits and so on, and it ranks these, And as I said, you may end up with one or let's say one or more edges that appear to be causal. And then in step 4, once you have a causal, an edge that has a significant Leo score, a Leo score above a threshold, then you carefully evaluate the fitting indices of these SEMs that I mentioned here, but there are many others. But even at that level, the evidence could be due to a SNP that, let's say you have multiple SNPs. It is still highly dependent on which SNPs you selected. And therefore, I suggest that in a step 5, you do a robustness analysis of your causal inference. So let's say here in this graph, I carried out a causal analysis based on 3 SNPs. But you could ask, well, what if I drop one of them? What if I add one of them? Ideally, you find evidence even if you drop some of these causal anchors. All right. So before I go to an application that was published, I wanted to now give you some R code. And so remember that this is actually part of the software tutorial that we started with in a previous lecture. Remember, these are the female liver example data from Jake Lucis. And we already covered how we found modules, how we do module preservation analysis. But one section in this tutorial, which we skipped, was called Systems Genetic Analysis with NEO. And so here I will just describe this approach. So by the way, why do we call it systems genetic? Where does that term come from? Well, systems comes from that aspect that we do a module-based analysis. Where there's somewhere is a WGCNA part of it. But why do we call it genetic? Well, because we have genetic data. And remember, we found this blue module that was related to mouse body weight, okay, and various other physiologic traits of interest. But also, we had over 1,000 genetic markers available from this F2 mouse cross, and therefore we could look for QTLs for mouse body weight, but also we could ask, well, which of the module genes relate to the same QTLs? So remember, in order to find QTLs or really SNPs that relate to mouse body weight, you can do a lot score analysis using the QTL-R package. And we described that yesterday. Brian Yandel described a wonderful R software tutorial for that. But also, you can really use a correlation test if you code the SNPs in this numeric fashion, and you can just ask, well, which SNPs have a significant correlation with body weight. And also, you can ask, by the way, can you see this? Yes, okay. You can ask for eQTL hotspots, meaning within a module. So you can ask, on the simplest level, you could ask, are there QTLs that affect the module eigengene? Alternatively, you could ask, among those several hundred genes in the blue modules, how many of them correlate with a SNP that also correlated with mouse body weight? And we refer to to SNPs that affect many genes inside a module as a module QTL. And for the blue module, we actually found 9 such module QTLs, but there was one QTL on chromosome 19 that was particularly exciting because not only did it affect a lot of genes inside the module, but also it had a significant correlation with mouse body weight. And therefore, we focused on this one SNP as a causal anchor of the entire edge orienting analysis. So now in this final step, which goes towards finding key drivers of this module, we then calculate these Leo scores. So what do we evaluate? Notice there's this causal model that we evaluate. So we have this one SNP on chromosome 19, and we ask for every gene inside the blue module whether it relates to mouse body weight in this causal fashion, right? The SNP affects the gene expression level, and the gene expression level then affects mouse body weight. And to fast forward, remember one way to read the tutorial is by always looking at output, you know. And so there is a function, an R function called Leo single anchor analysis, which does exactly what the name says. It calculates a Leo score for a single anchor, meaning a single SNP. And what you see here, I evaluate whether this causal anchor causally affects trait A, which is the module eigengene, which may subsequently causally affect mouse body weight. And when so the way to run this type of analysis is to just copy and paste the blue code, which I will explain to you in a second. But basically you copy it, you paste it into the R session, and what the R software produces are these Leo scores. And what do we see? They're negative. And so it means there's absolutely no causal no evidence that this blue module in its entirety causally affects mouse body weight. You know, so it's a negative finding. You know. Remember also in my previous lecture I mentioned there was another module right next to this blue module that also related to mouse body weight, and also the brown module showed no relationship to mouse body weight. So it's a negative finding. However, the module was comprised of hundreds of genes, and as I said, a more meaningful analysis perhaps is to restrict the attention to all the genes inside the blue module and to evaluate which of the genes in the blue module show a causal relationship with mouse body weight. And so now this R code evaluates now every gene. It will take a couple of minutes. But to already tell you, we then focused on 3 genes that have a Leo score larger than 3. Remember, this is on a log scale with base 10, so it's 10 to the 3. So if one of these genes is implicated, for example, this gene, which is cytochrome P450 family 2, for this gene there's strong evidence that it causally affects mouse body weight, meaning this causal model fits 1,000 times better than any of the 4 alternative models. You know. And so when we applied it to all the module genes, there were 3 hits, and only one of these probes was associated with a gene symbol. As I mentioned, this cytochrome P450 related gene. And actually our colleagues had found that this gene was related to body weight, and they just based their results on a QTL study. You know. So now it's interesting to produce a correlation table between all of the variables. I always like to look at correlations so that I get a sense of the effect sizes. You know. So remember, we started with this causal anchor, this one particular SNP, and we can just ask, well, how correlated was this SNP with the blue module eigengene? And the correlation is 0.2. I don't know how you look at it. If you are a GWAS person, that's a huge correlation, right? In any event, incidentally, this SNP also has a very significant correlation with mouse body weight. Again, that's a huge correlation if you have the perspective from a GWAS study. In any event, this SNP has a really extremely high correlation with this particular causal candidate gene. Remember how this probe, this corresponds to the cytochrome P450 gene. So this probe has a negative correlation of 0.70, So what does all of this tell me? If you really believe the SNP, if you really think this SNP affects mouse body weight, then yes, this particular gene is interesting in its own right because it already has a very strong correlation with the SNP, But further, apart from that, as I said, when you focus on this local or this causal model, then using this SNP again leads to a fit statistic that indicates the causal fit is really works well. All right. Are there any questions? Let me show you the output. So notice the R command finished, and here we evaluated 524 genes that were inside the blue module. And so out of these 524 genes, only 3 genes had a Leo score larger than 3. So they would be strong causal candidates if you assume that the causal anchor is appropriate and make various other assumptions. Now, some of you may be interested to learn more about the structural equation model R package developed by John Fox. And I just wanted to give you just some statements to guide your reading. So what you do, of course, you have to read the SEM package into R. And so you just do that by, for example, here copying and pasting. And now the other thing that is required when you evaluate these causal models is you have to specify the causal model. And so there is an R command within the SEM package called specify model. And here I have these 5 causal models, and for each causal model I need to specify it, and I do that. So this is causal model 1, causal model 2, and so on. And so here we use a certain formulation of causal models called the RAM formulation. But basically you say there's an edge from A to B. Remember, the causal model 1 assumes that A causally affects B. Then I tell it this will be the name of the parameter, the path coefficient, it's beta. And then you can specify initial values if you have, but here I say I don't know the initial value. So in any event, you get these 5 you specify these 5 causal models, and then the next thing is you need to calculate a model fitting p-value for each of these causal models. And this is accomplished by using this function model fitting p-value. And the function takes as input a structural equation model object. And then, as you notice, it makes use of the chi-square statistic and the degrees of freedom and calculates this p-value. And then there's another R function called Leo single anchor, which I used a second ago. And it, of course, evaluates. So this R function calculates these 5 different model p-values and ultimately defines the Leo score in the fashion that I described. It takes the log with base 10 of the ratio of these p-values. And so again, the purpose is to allow the data analyst to quickly prioritize thousands of causal models, right? Here I actually only had 524 genes, but sometimes you have 30,000 probes, and you could screen through 30,000 probes and say which of the 30,000 probes is interesting if I look for a causal one, you know. So that's the purpose. But again, the SEM object that we fit here, see here we fit this a causal model using the SEM command, and this object reports a host of fitting statistics by just running the summary statement, for example. So you would then carefully look at the genes that you found, whether they actually do fit things well. But never mind all the statistics. At the end of the day, what you really need is biological validation. So let's come back to the PowerPoint slide. And now I want to describe how we applied this type of approach to data for studying familial combined hyperlipidemia. And these are data that were generated by Päivi Päiväkanti, who was the PI on this study, and Chris Plaisir, who was the first author. So FCHL is, of course, a lipid disorder, and it's characterized by elevated fasting plasma triglyceride levels, total cholesterol, or both. And Päivi Päiväkanti had previously carried out a linkage analysis in a Finnish dataset and found one particular SNP that was highly associated with this hyperlipidemia. What does that mean for us? We really have a good causal anchor. That's what it means. There was strong evidence that it relates to hyperlipidemia. Now, this SNP was located in the 3' UTR of a transcription factor, this USF1, and Päivi Päiväkanti and her colleagues had used ChIP-seq data to show that, yes, this transcription factor mattered. And so in this study, then, my collaborators looked at 70 extremely discordant individuals. Now, remember how we earlier discussed what kind of sample size is needed to do a causal analysis. And I completely agree with the statement, ideally you want several hundreds at the very least. And that when you have a really good causal candidate, because if you don't, you want several thousand, you know. But in any event, let's just be courageous and proceed with the analysis. So we, to defend this data set, so these were extremely well-characterized individuals. Half of them were on the low end of hyperlipidemia, and half of them were at the high end. So these were extremely discordant individuals. And also, the other thing is we studied a relevant tissue. So these were fat biopsy samples, you know. But I do mention that a sample size of 70 gives you only 80% statistical power to detect a correlation coefficient of 0.33, right? That gives you a sense. Now, in order to make any sense of these data, we needed to apply a data reduction approach. And of course, WGCNA is a proven method for reducing the data. And so here we used the blockwise module detection approach to find modules in roughly 15,000 genes. And we ended up with 28 modules. And so you've seen this graph before. So these are the 28 corresponding module eigengenes. And here are the FCHL component traits. And remember how I mentioned to you there's this one module in the middle, the TAN module, that has significant correlations with total cholesterol, triglyceride levels, and importantly, also with the SNP that was implied in the linkage study. So that was a very interesting module to us. But of course, now one can look at hub genes in that module. But now, of course, we have this SNP, and we can do an exciting analysis. We can ask, is there evidence that this module eigengene is causally related to disease status? And so in other words, we can evaluate this type of a causal model. Is this particular SNP, does it causally affect the module eigengene, and does that causally affect some of these traits? And what we found is that the Leo score for the trait measuring fasting plasma triglyceride levels was 0.3, which is not terribly significant. What does that mean? Remember, it's a log transformed value. It just means that this causal model fits better, 2 times better than the 4 alternative models. Is 2 times better significant? Not really. So in our paper, we recommend for the single marker analysis a threshold of 1, meaning the model fits 10 times better. Having said this, this study was severely underpowered. There were only 70 individuals, right? So, and therefore we lowered the threshold. Incidentally, when you calculate the Leo score for disease status, meaning FCHL yes or no, the Leo score was 0.25. There is a faint evidence that this model may be causally related to the disease. Now, perhaps a more meaningful analysis is to carry out this analysis on a gene level, where you now look at all the genes inside this TAN module, and I'd ask, well, which of them are causally related, for example, to triglyceride levels? And it turned out that there were 171 genes that, for which we found some causal evidence for triglyceride levels. And there were 18 genes for which we found evidence that they causally related to disease status, Incidentally, often audience members ask me, can you use binary variables here, because, I just echo what other people in the field say. They say, yes, you can, with all the limitations, but people use it, So here we code binary disease status in a numeric fashion, 0 and 1.
Steve?
Yes?
Were the Leo scores significantly higher for the individual traits than the eigengene?
Yes. Yes, they were. Sometimes much more, think 1 or even more. I forgot actually what threshold we used for finding these causal candidates. I think we may have used 1, So in any event, we had these causal candidates, but now we're in a human setting. We really can't do validation experiments, knockout studies, and so on. So we do the next best thing. We look at literature findings, So here's a table of these 18 causal candidates. And in a second I showed, well, here I report some findings from the literature. And so many of these genes had been implicated. And Mark Keller in the audience may recognize some of them. Other people have never seen them, fair enough. I briefly mentioned there was also a GBOS study that has been mentioned now a couple of times by other people in human subjects by Kathirasan et al. And so what you can do is you can ask, are there SNPs that underlie these causal candidates, and were these SNPs implicated to be related to hyperlipidemia relevant traits in this GBOS study? If so, there's indirect evidence that yes, maybe they may be causally implicated. And so yes, here my colleagues report various p-values from this study, but there was one particular gene of interest, there was this one gene, the FATS3 gene, fatty acid desaturase 3 gene, and this was of particular interest because, again, we had found it, and also there was a SNP located within that gene that had been implicated in this GWAS study. And so then my colleagues focused on this particular SNP in the FATS3 gene and showed that even in this subgroup of 70 subjects from the Mexican population, there was evidence that this SNP related to disease status. So there was, again, some evidence that, yes, this FAT3 gene may be causally related. So just to remind you, why did we use WGCNA? It was really needed in order to leverage this small data set. It allowed us to find this very exciting module related to hyperlipidemia. We were able to use the KME, the module membership measure, in order to carry out a functional enrichment analysis for Go ontology categories. I skipped that. There's one slide where I mention the Go results. They made sense. We found lipid-related themes. And also, it allowed us then to restrict the causal analysis to the genes inside this module. And what I like about this study, it tells you that, again, this causal analysis applied to a module may provide an alternative way of arriving at these key driver genes. Bin Zhang described his approach for finding key drivers, but there are many other approaches for then looking at these sub-networks and prioritizing them. Again, the R software that I just presented a second ago corresponds to one of these book chapters. And here I acknowledge several of my collaborators, in particular Päivi Päijykanta and Chris Plaisir, who were the authors on this systems genetic analysis. Okay, thank you. Are there any questions? Yes? Yeah.
Do you know the distribution of the Leo score? I mean, do you have any empirical idea? Are they normally distributed?
No, I wouldn't make this claim. So the question is, what's the distribution of the Leo scores? I'm not sure. I don't think it's normal. I forgot what it looks like. It typically is negative. It's actually hard to find a Leo score larger than 1. That's what I would tell you. I often find that. Often I reanalyze somebody else's data, and I want to find causal candidates, and really nothing shows up. Yes. Other questions? Yes.
So for the adipose genes version data, when you applied WGCNA, did you use differential expression?
No. So when we applied WGCNA to these 15,000 genes, that is what we call an unsupervised WGCNA analysis. So these genes were prioritized probably based on the variance or the mean expression value.
So then when you go on to do a Go enrichment,
Yes.
do you restrict the Go terms?
That's a good question. So whenever let's say you start with a set of 15,000 genes, and then you want to do a Go enrichment analysis or David E's analysis, then the question is, what is the background list of genes? Strictly speaking, you should, of course, only use the 15,000 genes that you started with as a background, But many authors are lazy about it, and they just use all genes in the genome as a background, And I often follow that convention, what everyone else does.
But that's even less conservative than the question I was getting at, which is you found lipid-enriched genes, but that's not so surprising because you used adipose tissue.
Well, so the question is, was it surprising that this tan module was significantly enriched with lipid-related Go categories? I don't think it reflected that we focused on lipid tissue because there were other modules that were not enriched with these lipid categories. Do you see what I'm saying? Remember, we had 28 modules and not every module was enriched with lipid metabolism related themes, you know. Having said this, I agree with the gist of your comment. Sometimes it happens by pre-selecting genes. For example, if you look at brain tissue and you select genes that are overexpressed in brain tissue, then you find neuron-related themes, you know. So yes, that could go wrong, and so be aware of that potential bias. Yeah, other? Yeah.
Usually in genetics, and we do the dual association, we have like huge sample size, like 10,000 individuals.
Yes.
But when we do the gene expression, we usually just have a very small subset.
Yes.
10,000 individuals.
Yes.
I assume, in theory, this method can be applied to that kind of data. But can you comment on what do you think is the best practice?
Yes, so the question is that sometimes in genetic studies we are fortunate enough to have tens of thousands of samples, but for gene expression studies we may have a couple of hundred samples. And how to deal with that? Well, remember, we only need this very large sample size for GWAS studies because the effect sizes are so small. And so as a workflow, you could start with a very large GWAS data set to find credible SNPs. And then when you do this systems genetic approach based on gene expression data or DNA methylation data or other omics data, you really then focus on these causal anchors. Because the hope is that the gene expression levels or methylation levels show stronger effect sizes with your clinical traits, and therefore you get away with far fewer samples. And I think our applications demonstrate that. You know, sometimes a couple of hundred of subjects are sufficient. Even in this study by Pivy, if you are in the relevant tissue, human fat tissue, and study the trait, then we even as few as 70 samples allowed you to find highly significant relationships between a module and the hyperlipidemia-related traits. So yes, that works. Yeah. Okay. Yes.
Just to comment on your response to his question.
Yes.
If I have to do these genome-wide association studies, I will do in a reverse direction.
Yes.
The direction will be first find out the expression regulators or methylation regulators.
Yes.
And then use those limited number of functional SNPs to test on test their association with phenotype, because the phenotypes
Yes.
are most of the time complex as
Yes.
compared to
Yes.
those quantitative traits like expression
Yes.
or methylation.
Yes. Yeah, just to echo it, so another strategy is to start out with the omics data, the gene expression data, find gene sets or regulators that matter for the trait. It could be a module, could be transcription factors or what have you. And then look at SNPs that underlie these genes, for example, that co-locate with them in some shape or form. And then use this set of SNPs I want to call it SNP set enrichment analysis, for example, for in a GWAS study. And I really think that it's a very promising strategy. As a matter of fact, I use it myself, you know, so in some of my projects. Yeah. But with this, let me stop and remind you that this time lunch ends a little bit earlier at 1:15, so please be here because there will be another very exciting lecture from Bin Zhang. Yeah. Thank you. See you at lunch. Yeah.